Krebs cycle, also known as the citric acid cycle or tricarboxylate cycle. It is the reaction for most of the energy needs in the complex organism. It is used for generating energy through oxidation of acetyl-CoA. It is also used for synthesis of NADH and production of amino acids. The location which this process occurs is mitochondria, which are eukaryotic cell organelles. This activity begins with the pyruvate molecules that were generated in the cytoplasm during glycolysis. This pyruvate will enter the mitochondrial matrix to find coenzyme A.
Once pyruvate enters mitochondria, it will undergo decarbolization, in which oxidation occurs by existence of NAD+, and then it will attach to coenzyme A, generating NADH and CO2 in the process. It will later producing acetyl-CoA, which have two carbon compounds. Now we are entering the Krebs cycle. First step, citrate syntase enzyme will remove acetyl group from acetyl-CoA and combine it onto oxaloacetate, which have 4 carbon compounds, to form citrate with 6 carbon compounds.
So, for step 2, citrate is converted to its isomer, isocitrate. The enzyme econitase catalyzes this reaction. Step 3, isocitrate undergoes dehydrogenation and decarboxylation to form 5-carbon alpha ketoglutarate. A molecular form of carbon dioxide is released. Isocitrate dehydrogenase catalyzes the reaction. It is an NAD+ dependent enzyme. NAD+ is converted to NADH. Step 4, alpha glutarate undergoes oxidative decarboxylation to form acetyl-CoA, a 4-carbon compound. The reaction is catalyzed by the alpha ketoglutarate dehydrogenase enzyme complex. One molecule of carbon dioxide is released and NAD+ is converted to NADH.
Step 5. Succinate. Succinyl coenzyme A convert to succinate. This reaction catalyzed by enzyme succinyl coenzyme synthetase. Succinate have 4 number of carbons. In this reaction, GTP molecule is generated. Succinyl coenzyme A is high energy compound. The energy stored in this compound is used to attach a phosphate molecule to GDP, and this GDP will convert to GTP. GTP soon will convert to ATP because of the substrate level phosphorylation. In this step, GTP is formed. The last one, coenzyme A is released in this reaction. Succinyl coenzyme use is high energy compound and its coenzyme to be converted into succinate. We move to the step 6, which is fumarate. Succinate convert to fumarate. In this reaction, it is catalyzed by enzyme succinate dehydrogenase. Because its name is dehydrogenase, it is remove hydrogen from succinate. The hydrogen that is removed is accepted by FAD. FAD is converted to FADH2 and succinate is converted to fumarate. FAD is used here as it requires less energy change to get reduced to FADH2. fumarate has 4 number of carbon. It is the only enzyme that located on mitochondria inner membrane.
After stereospecific hydration, fumarate catalyzed by fumarase is transformed to malate. The last step of NAD couple, oxidation of malate to oxaloacetate is catalyzed by malate dehydrogenases and close the cycle. Through the Krebs cycle, following products are generated: 3 compounds of NADH, 1 compound of FADH2, 1 compound of GTP, 2 compounds of carbon dioxide. Now, since the glucose is split into 2 pyruvate compounds, for each molecule of glucose, this cycle runs twice. Hence, the product form are 6 compounds of NADH, 2 compounds of FADH2, 2 compounds of GTP, and 4 compounds of carbon dioxide.